Hey everyone, welcome to chapter 25. So, in chapter 23, we talked about genetics, Mendelian genetics, and in that chapter, we learned that there was something that was being transmitted from parent to offspring. So, parents were able to give their offspring some characteristic, but we didn't really know what that thing was in the 1800s when Mendel was doing his research. It wasn't until the mid 20th century that we really figured out that the characteristic, the factors, were genes on DNA. At this time, or prior to this time, let me just say, we didn't know if it was DNA. We knew there was something called DNA now, but we didn't know if it was DNA or if it was protein that was actually the factor that was transmitted. In the mid 20th century, geneticists were just determining the structure of DNA. They were determining what the function of DNA was, but they weren't 100% understanding of any of it. So what did we know? We knew that genetic material, so something was able to be stored in our cells. We knew that. And this information coded for development, structure, and metabolism. We also knew that it had to be stable so that it could be replicated and transmitted from parent to offspring. That's what we knew. So look at the date in the 1930s. So this is very young. A scientist by the name of Frederick Griffith was studying Streptococcus pneumoniae, and he noticed that there were two types of Streptococcus pneumoniae. One was called smooth Streptococcus pneumoniae, or he called it the S strain because it had a capsule that made it smooth. The other was called the rough Streptococcus pneumoniae, or the unencapsulated, so it was a rough strain, okay? And if he injected Streptococcus pneumoniae into rodents and it was the S strain, the rodents got sick and they died. And he was able to find, when he looked in their blood, he was able to find these smooth Streptococcus pneumoniae. If he injected the rough strain of Streptococcus pneumoniae into these same rodents, the rodents survived. They did not die. And if he then killed them and looked for the bacteria, he did not find the bacteria. So then he thought, okay, the capsule is the difference between this and this. Otherwise, there's nothing different. So the only difference between this cell here and this cell is that this cell has a capsule around it. So maybe it's the capsule that is pathogenic. So then what he did was he killed the bacteria by heating them up. And then he injected those heat-killed bacteria into more rodents. And the rodents did not get sick or die. And he thought, well, this doesn't kill, or this kills. This one here doesn't kill. This one here doesn't kill. What if I take both of these that don't kill and infect or inject them into a rodent together? So the capsule itself doesn't do anything, and the rough strain doesn't do anything. So he mixed these together and injected them into the mice. And what he got was a bunch of dead mice. And then when he looked into their blood, he found live smooth. So you have dead smooth, live rough. And what he found in the mice was live smooth bacteria. So what was happening? This process he called transformation. And so basically, bacteria can take in DNA from an environment as naked DNA. So bacteria take in DNA as naked DNA in solution. SOLU, not OSOLO, sorry. So basically, these bacteria here were able to take in the DNA that was leaking out of these dead cells, and they would take in that DNA, and then they were able to produce their own capsules, which is what caused the S strain to be seen. And so this process occurred before we really knew if DNA was the transforming principle. He just said there is a transforming principle that allows bacteria to take in some genetic information so that it can then, from dead cells, so you take in genetic information from a dead cell as naked genetic information, and you can transform yourself. That's basically what he was saying. And we know today that that's DNA. Now, they did not know this in the 1930s. In the 1940s, we learned about genes, and genes exist on chromosomes, and chromosomes contain both protein and nucleic acid. So there was a huge debate over, is DNA or is protein the genetic material? So a lot of scientists thought protein was the genetic material, and here's the reason. Proteins are what drive all of our metabolic reactions. Proteins are what form all of our structures. Proteins, so if you look at a person, you're looking at protein. Skin is protein, eyes are protein, muscles are protein, hair is protein, nails are protein. So protein is really important. We obviously know this. And so why wouldn't protein be that genetic information that we are transforming, or that we are transferring from parent to offspring? But we weren't sure if it was protein or DNA. DNA could have been the other option, right? And so in 1944, Avery, McLeod, and McCarty ran an experiment, and they reported that DNA was the transforming material. And so here's what they did. They looked at the exact same smooth cells. They looked at the exact same smooth cells and rough cells that Frederick Griffith was looking at. So he killed smooth Streptococcus bacteria, pneumonia bacteria. They had it in a solution. So these are dead cells. They then added to the solution, so there was 3 different tests going on. They added proteases. So these are enzymes that will break down proteins. They added to another tube RNases. These will break down RNA. and DNA aces. These will break down DNA. They then took the solution and they mixed in the living rough cells. So remember, if you have smooth cells and that are dead, and you take rough cells that are living and you put them together, you know that you're going to get smooth living cells. So they figured, well, if protein is the transforming principle, then if we break down all the proteins, we will not produce those smooth cells. But they did. So they knew, okay, protein was not the transforming principle. In the RNA experiment, if RNA was the transforming principle, then breaking that RNA down would lead to not being able to produce those smooth cells. But again, smooth cells that were living appeared. RNA was not the transforming principle. So now they took DNA and they added the DNA ace. So this is going to break down any DNA. So now we have no DNA. If DNA is the transforming principle, then the R cells, those rough Streptococcus pneumoniae, will not be able to take in the DNA, and you will not produce those smooth cells. And lo and behold, without the DNA, there was only rough cells able to be seen. So this is where we figured out that DNA, not RNA and not protein, was the transforming material. But still, there were people that were not 100% on board with the fact that DNA and not protein was the genetic information that was being transferred from parent to offspring. And so in the 1950s, Alfred Hershey and Martha Chase, they ran an experiment, and what they used were bacteriophages. So a bacteriophage, bacteriophage, is a bacterial virus. A virus is a bag of nucleic acid, basically. So I'm just going to kind of put this circle, and bacteriophages are not circular, but I'm just going to draw it this way just to show you. And so inside is nucleic acid, okay? On the outside is a protein capsid. So that's kind of the idea of what a bacteriophage looks like. So what they did was they used radioactive phosphorus in one experiment. Radioactive phosphorus, nucleic acid, has phosphorus in it. So they basically tagged the nucleic acid in this experiment. And then in the second experiment, they used radioactive sulfur. And so radioactive sulfur is in the protein capsid. So hold on one second. That was my dogs going crazy because somebody was walking by. I apologize for that. So in the second experiment, they used radioactive sulfur to tag the protein capsid, okay, in this experiment. And what they found was that what actually enters the bacteria is DNA. And so let's look at that experiment really quickly. So here we have the viral DNA is what is tagged. And so it's what's going to glow because it is radioactive. And so this is what a bacteriophage looks like. I told you it wasn't circular. It looks more like a weird spider almost. So the virus itself attaches to a bacterial cell, and then it injects the DNA into the cell. That injected information then is able to be replicated with the cell, or it can replicate on its own and cause all kinds of problems for the bacteria. In this case, the bacteriophage did not kill the cells. What it did was it was injected into the cells, right? And now the bacteria have that viral DNA inside. So if the DNA is in fact the genetic information, it's going to be found inside of the cells. So after they allowed the bacteriophage to infect the bacteria, they then used a basically a blender to separate or to mix everything together, right? They then centrifuged the material, and so anything in all the cells would fall to the bottom because they're heavier. All the liquid is going to be on top, okay? What they found was that the radioactivity was in the bottom material with the bacteria, so down here, right? In the other experiment where we have the capsid is what is radioactive, again, we have the bacteriophage infect the cells, and the DNA enters the cell. We agitate everything together so that the viruses are dislodged, just like up here, and then we centrifuge. Now, if the radioactivity was in the cell again, then we would have to say, okay, that's the protein must be what's infecting and what's going into the cell and changing them. But in this case, it was the liquid up top that had the radioactivity, because this viral capsid doesn't actually enter the cell. It doesn't go in. Only the DNA went into the cell. Therefore, now we know DNA is the transforming principle. DNA is the genetic material. And so this has been a very long video, so I'm going to stop here, and we'll get into what DNA looks like in our next video. Bye.